So, okay, that's the idea. So it was a great seminar. We learned a lot about Trinity at that time. Let me see here if we see Marcelo Brandão here now. All right. Is anybody that is not in Brazil besides Elisa and Zhiping in the audience? We had advertised in some programs abroad. Just curious to see. I think that we only have Brazilians here. All right. So we decided to have a chairwoman today, and thanking Elisa for introducing me to people in UMass. She was a student. She was my student here since the first day of the undergrad course, and today is the second postdoc. So she's representing all the students in this range today, and she's on the command. So Elisa, let's go.
All right, thank you, Miguel. So hello, everyone. It's a pleasure to be here today, and especially after being away from Brazil for so long, even though part of me feels like I never left Miguel's lab, where it all started. And even better is that I have the pleasure to introduce you to Dr. Zhiping Weng, who's an amazing scientist that I have great respect for. And I had the honor to be part of her department in UMass for many years during my postdoc. And like she herself was explaining, since the very first pilot phase started in 2003, she has dedicated herself to the ENCODE project. And more recently, during phase 3 and phase 4, she served as the leader for the Data Analysis Center and coordinated the massive effort involving so many other labs, which has provided all the rest of us with a comprehensive data resource and some incredible insight into the functional genome. Her own group at UMass has been instrumental both in developing tools and integrating and interpreting this array of genome-wide sequencing data in order to identify and characterize regulatory elements and discover principles of gene regulation in mammalian genomes, as well as key features linked to human disease. So I hope you're as excited as I am to hear more of her recent work on building a registry of regulatory elements. And without further delay, I give the word to Zhiping. Welcome, Zhiping.
Thank you so much, Elisa, for the very kind introduction. And Elisa was part of our program for a few years, and it was really our fortune to have her. And we shall miss you quite a bit. Even I think Manuel is a bit at a loss now, and I hope he recovers. he was after you left. I think he was in a bad mood for a bit. Okay, so we really need people like you as part of our team, and I think even though you were not part of Elisa, you were not part of the ENCODE, but you were part of the GGR, right? You produced quite a lot of data. And
yeah, we did a little bit on genome regulation too.
Yeah, and then those data sets show up on the ENCODE portal when I search.
Oh yeah, we submitted for ENCODE because of the GGR connection.
Exactly, on the ENCODE portal. So I'm going to share my screen. Can you see my screen?
Yes.
With the talk, hopefully. I have 3 screens here.
All good.
So just want to make sure the correct one is on. So today, I'm very pleased to have the opportunity to talk to you about our work as part of the ENCODE Consortium. And this is part of the ENCODE 3 work, phase 3, as Elisa just mentioned. It has been published. That's why I'm talking about this work. I just want to mention that it has also been extended to ENCODE phase 4, which is what we are right now. So the registry of candidate cis regulatory elements, which we built using ENCODE 3 data, are being updated with ENCODE 4 and also GGR data in the meantime, and we're continually to provide newer updates on our resource, both at the ENCODE portal and also on our own visualizer called a screen tool, which you will hear in a minute. So let's see if I can move it. So a bit of the background of the human genome. So if you look at the human genome as this entire circle, it might be known to you already, but still is surprising every time I think about it. Less than 2%, actually roughly 1.5% of the human genome are those exons that code for proteins. And the remaining genome has all kinds of other things. We tend to have very long genes with long introns. So a lot of it is introns. And we also have non-coding RNA genes, a bit more than protein coding genes, maybe twice as many. And then we have pseudogenes. These are genes in the genome that no longer work. It's like remnant of previous protein coding genes. We have lots of repeats and transposons, most of them dead, but recording the history of transposon invasion. Among all these, and the category that I want to talk about today is the regulatory elements. These are bits and pieces of the genome that has the function of trying to regulate the protein coding genes. How are they transcribed in a temporal, which is in terms of time, and the spatial, which in terms of tissue and cell types, and location in our body. So the transcription of the genes are controlled by these regulatory elements. This is a class of the regions in our genome that's not very well understood, much less well understood than the coding exons. Even the estimate as to how much of our genome is devoted to regulatory elements, the estimate varies, depends on who does the estimate. The ENCODE itself has 2 papers and gives out different numbers here. So that just tells you that it's not as well defined, not nearly as well defined as protein coding genes. So I want to give a few examples as to what kind of regulatory elements I'm talking about. So here would be a typical gene with, say, exons, introns. Introns are not drawn to scale. Usually the introns of our genes are much, much longer than exons. And the gene is going to be made into an RNA transcript. And the transcript will be spliced and then translated into proteins. All that you probably already know. So the gene is here. The gene itself is like, you know, a recipe. So, and the transcript is perhaps a cake. And when do you want to make this cake is what regulatory elements tells you. So here are some examples. One is the promoter, which is a region where the transcriptional machinery and the activators would dock themselves and tell the gene to be transcribed. Okay, this is usually immediately upstream the gene. The black arrow here indicates transcription start site, TSS, which you would hear shortly with the acronym TSS is this black arrow. And usually promoter is the region within 2 kilobases, 2,000 base pairs, right upstream the TSS. And enhancer are these, as the name implies, are regions in the genome that activates the transcription. It helps the promoter. So enhancers, they enhance the promoter. And they bring in additional transcription factors, interact with the RNA polymerase to promote the transcription of the gene. There are also negative players, such as silencers or repressors. instead of enhancers, they enhance. The silencers, they silence and repress the transcription. They also work with the transcriptional machinery, basically that's the negative work. There is also one kind of element called insulators. The interesting thing about our genome is it's like a string that has unclear, it's just a mystery how the cell reads our linear genome, because it's difficult to tell where things start, where things end. So if you have an enhancer here, does it enhance this gene on its right or this gene on its left of this linear string, or both genes? And in what condition, during what time? So insulators are like the blocks that divides our linear genome into neighborhoods, so that enhancers do not accidentally activate the wrong gene. So it blocks interactions with nearby genes. So if you do something to mutate the insulator, for example, and then the enhancer has been often shown to spill its influence further afield and start to inappropriately activate the wrong genes. Okay, so these are probably the most well-known examples of regulatory elements. And why is ENCODE, or a lot of people, interested in annotating this kind of regulatory elements? There is a very practical reason. If you study the human population, people with different kinds of diseases, if you look at, if you sequence their genome and trying to figure out what's going on with them, why do they have these kind of diseases? And besides those coding exons, the exons, sometimes you will see they are mutated, especially for Mendelian diseases. Okay, usually if you study these people's genome with diseases, you find that mutations are highly enriched in regulatory elements. Okay, these examples I give, much more enriched in regulatory elements than the rest of the 98% of the non-coding genome. So here are some examples of diseases with well-characterized regulatory elements being mutated, such as polydactyly. This is diseases with less than 5 digits on your hand, cleft palate, and congenital heart defects. These diseases all have mutations in their regulatory elements. And depends on the estimates, more than 80% of the non-coding genetic variants associated with human diseases are in non-coding regions of the genome, and they are highly enriched in these regulatory elements. So, so that's why ENCODE, as a big consortium, has decided to set its goals to cataloging all functional elements in the human and mouse genomes, mostly human genome. And the consortium is very much in a resource mode. We want to develop freely available resources for the research community that's freely available to everybody, no strings attached. And as soon as the data are produced, it will be put it on the ENCODE portal so the community can download and use the data for their own research. And we primarily study human, but in the history of ENCODE, we have also studied other model organisms, such as mouse, fly, and worm in the past. But right now, it's mainly 90% of ENCODE data on humans and 10% on mouse, roughly speaking. So here is one of the pictures before the pandemic, where people can aggregate in such big numbers. One of the ENCODE 3 consortium meeting, and the ENCODE consortium has roughly 500 people. I know it pretty well is because we managed the authors list for the ENCODE 3 publication last year, and that's how many authors we have.
You are in Salk Institute there, right?
Yes, Miguel, that's exactly right. That's the Salk Institute. That's where the sunrise and sunset somehow aligned. It was beautiful, right?
Mm-hmm.
And right now, this would be illegal, I assume, during the COVID. But that was a very good meeting. Yeah, it was Salk. You have sharp eyes. So the project, how does the project get organized with so many people? So there are different components. There are data generation centers. There is a data analysis center, and then there is a data coordination center. As Lisa just mentioned, I'm heading the data analysis center for ENCODE during phases 3 and 4. And we have roughly 8 labs in the data analysis center, and all pretty with good expertise in computational analysis and data integration. So, so today I'm going to talk about the ENCODE 3 publication that we made quite a sizable contribution to. So this was published last year during the July 29th issue. It's a package. So this is ENCODE phase 3, and then you can see there's a number 3 over here, and presumably these wires, it's like the DNA of our genome. And so here, this is the main paper. There are a few, quite a few companion papers in the same issue. If you're interested in, go and look at the whole issue. And this main paper describes ENCODE data and also the registry of cis-regulatory elements that I'm going to tell you about today. So these cis-regulatory elements, we kind of focused on promoters, enhancers, and insulators, which I mentioned earlier as the cis-regulatory elements. And let me go a little bit about the kind of data we used to annotate these elements. So if you look at an enhancer, it's very commonly understood that enhancer is a region in the genome that has displaced a nucleosome. So what is a nucleosome? So our DNA in our cell, in all eukaryotic cells, it's very long. It's a very long and skinny fiber. So for example, for humans specifically, we have 3 billion base pairs of DNA. And we actually have 2 copies of it, one from our mom, one from dad. And if you line them up head to tail, it amounts to 2 meters long, which is very long, longer than our height. So how can we package such a long DNA in fragments? We have 21 chromosomes, autosomes, and then 2 sex chromosomes. So they're in fragments, nonetheless, very long fibers. It's also highly charged. So they're going to repel each other because the DNA is highly charged. It has the phosphate backbone, negatively charged. So what the cell does is it wraps around the DNA, which is showing this line here, around something called the histone proteins. These are also highly charged, kind of compensates for the DNA's charge. So these are histone proteins. There are 4 kinds here: histone 2A, 2B, histone 3, and histone 4. Each one has 2 copies in this histone complex. So it's called an octamer, has 8 of them, 4 kinds, each has 2 copies. And this is a histone octamer wrapped like roughly 2 turns by the DNA, and then this complex is called a nucleosome. So most of the DNA in our cells are packaged this way so that you can fold a 2-meter long fiber in segments into the tiny nucleus in every single cell in our body. And when you have a highly compacted nucleus, it's like a closet, right? If you want to put away your, for you right now, it's summertime. You want to put away your winter clothes, you put them in the closet. And when the winter comes, you need to take it out and unfold so that you can use it. So it's the same kind of logic. So if you look at the human DNA, if you package it away, usually it's quiescent. You don't do much with that piece of the DNA. But if you need it to transcribe a gene or to regulate the transcription of the gene, you actually unfold it and release the histone octamer here. Normally it's here, and then now it's gone, and so that you can get access to the DNA. And when you need access, it really means is these transcription factors. These are regulatory proteins. They need to come and recognize the DNA so that they can, normally that's what we say by transcription factors bind to the enhancer. And then they can go and talk to the polymerase, the transcriptional machinery, to turn the genes on or off. So, the probably a pretty accurate picture of an enhancer is a region of the DNA that has lost the histone octamer, but in its place, now it's bound by a bunch of transcription factors so that you can activate or repress, or in the case of repressor, or insulate, in the case of insulator, the gene regulation. So in order to find these kind of regions in the genome, now we can use this characteristic to find it, okay? So one possibility is we can use antibodies. The Y-shaped molecule here indicates an antibody that recognizes these transcription factors. So ENCODE has 2 data producing centers who do these kind of experiments. They generate thousands of data sets using antibodies, using chromatin immunoprecipitation, and then sequence the DNA that's bound by a transcription factor and put it genome-wide. They show up like these kind of peaks on a genome browser. I just want to add one more sentence here about these transcription factors. Out of the total 20,000 genes, protein coding genes in our genome, actually 1,500 of them code for various transcription factors. 1,500 out of 20,000. So that is quite a big number of array of TFs. Here we show with 3 different colors. They have cell type and temporal specificity. They work together to do gene regulation, perform gene regulation. And also, you can recognize some kind of histone modifications. And these nucleosomes flanking enhancers and other regulatory regions, for the histone proteins, their N-terminus tails very often are modified, okay? So they are modified covalently. So methylated, acetylated, ubiquitinated, and different kinds of modifications indicate different kinds of functions for the region. So ENCODE has also one data producing center making a lot of ChIP-seq data sets for histone modification. And here is a specific one, H3, histone 3, lysine 27 acetylation, which is a marker for active enhancers. And also, the property of this region that doesn't have a histone here can be explored. So if it doesn't have histone proteins here, then it becomes more accessible to endonucleases such as DNase 1. So you can treat the cell nucleus with DNase 1 lightly, and you will cleave the accessible DNA, and then you can sequence the DNA. And when you map to the genome, you see these kind of peaks as well. And those correspond to all kinds of regulatory regions. So ENCODE has one producing center that makes lots of DNA-seq data. So here is an example of these kinds of data in one particular locus. This is SP1, is a transcription factor gene. And you can see here with exons, introns, exons, long introns here. And you can see that at the promoter region, you have open chromatin, which means the histone is being kicked off. You're missing a nucleosome. So you have a peak at the promoter. And flanking this peak, the neighboring histones have the appropriate modification. So here is histone 3, lysine 4 trimethylation. It's a known mark for histone mark for active promoter. And this is the other histone mark, which I mentioned before, histone 3, lysine 27 acetylation. And it's a mark for active enhancer. And you can see these regions, they line up really well. So you have a missing nucleosome in the middle, and then marks on the flanking nucleosomes. And also, these are far away enhancer regions, similarly has a nucleosome missing, and then histone marks on the flanking nucleosomes. And I want to mention CTCF. CTCF is one of the 1,500 transcription factors. And it's a very special one because it is the only transcription factor that binds to insulators in our in mammalian genomes. And it also has additional functions besides being an insulator binding protein. It has important functions for organizing the chromatin three-dimensional structure, forming loops and the domains. So ENCODE has a lot of CTCF chip-seq data. And I just want to point out in CTCF binding, very often you also see open chromatin signal from the DNA signal, DNase data. Okay, so with available data in ENCODE, we have tens of thousands of genome-wide data sets. So the Data Analysis Center was charged to come up with a method for predicting regulatory elements. This is not a new charge because there have been a lot of efforts in the field and also within ENCODE groups for predicting regulatory elements. And so we could use one of these methods. Some of them are actually quite sophisticated, machine learning, different kinds of approaches. They are being applied to ENCODE data, and the results of these methods are being made available on the ENCODE portal as well. But the Data Analysis Center thought about it and decided that because the ENCODE data matrix is quite sparse, and we want to develop a method that stays really close to ENCODE raw data and can be applied across as many cell types as possible. So we came up with actually a rather simple method, and that's what we did for the registry of CCREs. So I just want to walk you through our approach step by step at a very high level, so you can see why we did what we did. So we decided to anchor our registry of elements on open chromatin regions using DNase data. So here is each row is a DNA data on a specific sample. And you can see that they line up pretty well across the samples. Here on this slide, there are like 24 samples, and every row is a different sample. And you can see these open chromatin regions, they are pretty ubiquitous. They are observed across almost all of the samples. And some other regions are more cell type specific. They show up in some cell types, but not in others. Okay, so when you take all of the DNA's data across all the cell types that we have in ENCODE, right now we have like 1,200 such data sets. What we did was for each region, we picked the strongest signal peak. We call these DNA's hypersensitive sites, in short, DHSs, DNA's hypersensitivity sites. So for each region, we picked the strongest DHS to be the representative DHS at the bottom here, black box, and to integrate across all DHSs. So each biosample has roughly 2 million DHS, and we have 1,000 samples. So you can have 1,000 times 2 million if you don't condense them. But after we condense them into individual location, in the ENCODE 3 paper, we used roughly 700 DNA data sets and curated 2.2 million representative DHSs in human genome 38 and 1.2 million RDHSs in mouse genome version 10. So the reason that we have fewer of them for mouse than for the human is because we don't have as much data for the mouse. We expect the mouse to have roughly the same number of regulatory elements as the human. So once we have a set of 2 million or so RDHSs from the 60 or so million in individual data sets, now we can annotate regulatory elements, which we call them candidate cis regulatory elements, CCREs. It's a very long name, but we used to call them CREs. And when we submitted our manuscript to Nature, one reviewer pointed out that because these are based on epigenetic signals and we don't have any functional data, we really have to call them as candidates. So we put another C there. It's a bit hard to pronounce, but that's what they're called in the paper. So we then take the RDHSs and promote a subset of them to be CCREs if they are supported by high DNA data and one of these 3 data types in at least one ENCODE biosample. So the reason we want another support is because these 3 markers really indicate promoter, enhancer, and insulator activities. And we want another line of support to give us some prediction of functional data. So at the end, out of 2.2 million, we have roughly 900,000 human CCREs. And out of the 1.2 million RDHS in mouse, we have 340,000 mouse CCREs. So that is at the end our registry. So for the registry, let's just look at the composition in humans. You can see that we further annotate them as CCREs with promoter-like signatures. And there are roughly 35,000 of them. These match up pretty well with the protein coding genes. Some of them are link RNA genes in the human genome. And we have enhancer-like signatures, roughly these can be proximal or distal to transcription start sites. So TSS stands for transcription start sites. Okay, so these are proximal ones, 142,000, and distal ones, there are more of them, 600,000. And we have some elements that are possibly novel promoters or poised enhancers. They do not overlap transcription start sites. They have some kind of signature, which is difficult to cleanly slot into these two kinds. So we gave it another category, and there are not many of them. And then we have CTCF only. These are good candidates for boundary elements and insulators. And there are 50,000 of them in the genome. So this is the whole collection in the human genome. And once we have the collection, we can further annotate them using ENCODE data in specific cell types. So here is an example of a hepatocyte. Hepatocytes are a cell type in the liver. And you can see these are all the CCREs. And we color code them consistently. Red indicates that it might be a promoter. Yellow indicates that it might be an enhancer. And then blue indicates that it might be an insulator. And green is universally used for open chromatin signal. And the gray areas are the CCREs that are in the collection, but don't have signals in this particular cell type. And so you can see that even though the total is roughly 900,000, there are only 137,000 being annotated in hepatocytes. And they fall into these numbers in the hepatocyte cells. And you can see the red corresponds to DNase plus K4ME3 and also K27AC. These two are highly correlated. And then the yellow, this is an enhancer candidate, corresponds to high chromatin accessibility and the high K27AC. And these blue ones are insulator candidates, correspond to open chromatin and also CTCF binding. There are several advantages of our particular approach for defining CCREs, I want to point out. Our elements have very high resolution. Each CCRE is between 150 to 350 base pairs long. This is because the DHS, the DNA signal, has very high resolution, and it corresponds to the length of a particular nucleosome that has been liberated so that the transcription factors could bind, as I mentioned earlier. Usually a human nucleosome plus a linker is a bit between 200 and 300 base pairs long. So you can see that 3 individual CCREs here correspond to 3 peaks in the DNA's signal. And the second advantage is the boundaries of these CCREs remain constant across all the biosamples. So they actually do line up pretty well, which provides a common reference and anchor to perform all kinds of integrative analyses. And just like genes in the human genome, we also accession the CCREs so that every release of the registry will refer to the previous release. So if you were to use one of these CCREs in your previous publication, you can always find them again and see what they have been updated to in the next release. So we consistently accession these elements using a human versus mouse and then genome version. And then these are unique identifiers, so you can find them. And also, as part of the ENCODE 3, we have built a a specific visualization tool called a screen, and you can visualize all individual CCREs and the underlying data in the screen using search and other tools. So let me see if I can, maybe I need to automatic. So here is a movie of how screen works. You can go to screen.encodeproject.org to access this tool, and it's real time. This is how fast the tool works, and you can search by cell type. And for example, you're interested in heart, all the biosamples that ENCODE has surveyed will show up. And once you get into a specific biosample, you can look at the signals specifically in that sample, and you can threshold it so that you have a shorter list. Change the threshold again, you will respond in real time. And you can look at the expression of neighboring genes. So ENCODE has performed a lot of RNA-seq experiments. And you can sort them by height of the expression. And this is a rampage assay, also part of the ENCODE production, that tells you the activity of individual transcription start sites. And you can loop through the different transcripts for the same gene. So you can play around with it after the talk, if you like. A few highlights I want to point out. So each element here, each row is element. Once you get into the element, this is the element accession. It tells you the region in the genome, and it tells you what kind of element we predict it to be, and the supporting signals from these 4 assays in each of the biosample. And you can go into the details about how many ENCODE data are available for this element. And you can go directly to the individual data sets here. And here is the gene expression, which I mentioned. If this element is near a promoter which is very close to the transcription start site of a gene, then you can look at the RNA-seq data to see where this gene is expressed. And also, we provide several data sets in ENCODE to link individual cis elements with genes that they regulate. When these ones are distal elements, they are far away from genes. And there are multiple ways that you can link them. For example, here are 2 methods that you can link distal elements with genes using chromatin, 3-dimensional chromatin interactions. 2 kinds of assay that ENCODE produce data on. One is the high-C assay on chromatin, 3-dimensional chromatin interaction, and also the CHEA-PET assay also produces this kind of information. And another is a single cell CRISPR-QTL method. QTL stands for quantitative trait loci, and this is a single cell method. And then also eQTL is expression QTL. You can perform these kind of experiments, also link CCREs with genes. So whatever data available, we incorporate them into screen. And here are the available data that when the slide was made, and we keep on adding new data as time goes on. So for example, if you look at this particular element, it shows what kind of data supports its linking with genes. And these are the genes that the element is linked to. So, okay, So that is an overview of the registry. So the next thing we did for the effort was to verify the accuracy of the registry. And this is one kind of assay. This is a promoter assay that you can use to verify the accuracy of the CCREs. So if you look at what this SURE assay identified in the whole genome, it's only a very small portion of the genome. And the overlap with the CCRE has an enrichment. But if you go to the specific cell type, this is K562 cells for which this assay was performed, you can see the enrichment is much more improved. And it's a promoter assay. That's why the enrichment is the most for promoter-like signatures, and it's less for the other kinds. And we also performed validation using experiments directly as part of the ENCODE project. These were experiments performed by Len Panakios group, and they take the region that's predicted to be an enhancer CCRE. They cloned it into mouse embryos, and then to see where the reporter shows up. So here is heart, this is forebrain, and then this is limb. So these are the blue color leg Z staining that indicates the activity of the element. So this is just a magnified image of the limb. So as part of the ENCODE paper, we actually generated 150 these kind of experiments, and these are the results to show that the validated elements. And here is a bar plot that shows the percentage of validated elements in 3 mouse brain regions: midbrain, hindbrain, and limb for one time point, and then forebrain, heart, and limb for another time point. And for the last few minutes, I want to showcase a use case, what you can do with the registry of elements. I started the talk by motivating our work by saying a lot of the mutations or variants that are identified in the human population that are associated with diseases are enriched in regulatory elements. So NHGRI, EBI, Genome-Wide Association Catalog is a collection of these kind of genomic variants. And it has over 3,000 studies, and it identifies these variants, very often single nucleotide polymorphisms called SNPs that are associated with all kinds of phenotypes and diseases. And due to the genome structure of our genome, not all the regions are completely recombined. So there is this concept called linkage disequilibrium. These are the variants that are linked on our genome, not recombined efficiently. So if you take these variants, and the variants that are assayed are called tagged variants, and then the ones that are linked with them are called LD variants. We actually took the entire collection of over 3,000 studies to identify what kind of enrichment the CCREs have with these SNPs. And you can see there are some examples here. For example, this is cholesterol, people who have high cholesterol. Their variants are highly enriched in the CCREs that are functional in liver. And you have lupus, which is an autoimmune response. Their variants are highly enriched in the CCREs that are defined in B cells, so on and so forth. So just as one example, we looked at the psychiatric disorder called schizophrenia, and it's particularly difficult to study. Two of the studies identified these SNP that's associated with schizophrenia, and it's shown here. And the interesting thing is this SNP does not overlap with an annotated element. Okay. However, it's in high linkage disequilibrium with 39 SNPs. So all linked with the SNP. And 6 of these LD SNPs overlap a CCRE. So we follow up with it. One of these 6 has high epigenetic signals in brain-related cells, which is shown up here in neural cells. And we further looked into all the samples we have in humans. This is DNA signal for human, and also in mouse. A lot of the brain development is conserved between human and mouse. And we can see that this element has a high K27AC. Remember, this is the enhancer mark. And throughout development, these are embryonic days in 3 brain regions. And we further performed, Len Panakios lab performed these transgenic mouse experiments, take that region that's a CCRE, and the reporter gene shows up in 4 brain regions being active. And the region overlaps a transcription factor motif. SP3 is a transcription factor, and this is the SNP, which alters the binding preference for these particular SNP. So just as example, as what you can do with the registry, if you have a variant, you can see which element it overlaps with, and then use the whole panel of ENCODE data, trying to narrow down its cell type specificity. So that's pretty much what I want to say today about the registry of CCREs. And let me just summarize what I have said. So we integrated thousands of epigenomic data sets to annotate almost 1 million candidate cis-regulatory elements in the human genome. And we validated these predictions, they are candidate cis-regulatory elements, using published functional data and by performing transgenic mouse enhancer assays ourselves as part of the ENCODE. And I show some examples about how these CCREs can be used to generate hypotheses for many different kinds of biological problems, including annotating disease-associated genetic variation. And as part of ENCODE 4, and we are continuing to improve the registry of CCREs in several areas. For example, by increasing the coverage of biosamples, using cycle-ENCODE data, using blueprint data, using public data. And we are additionally defining new types of CCREs, such as silencers, repressors that I mentioned at the beginning, currently not defined in the registry. And we are subsecting the enhancers into subtypes. And we're looking at cross-functioning, perhaps promoters could have enhancer activities as well in some cell types. And finally, let me conclude my talk by thanking the people who did a lot of the work. From my lab, obviously this is a ENCODE consortium effort, and all the data were generated by ENCODE producers. So we thank them for their effort of generating the data. The data analysis was done by the Data Analysis Center, which includes my lab. And these 3 people are my heroes for performing a lot of the analysis I told you today, pretty much all of the analysis that I presented today. Jo Moore was a graduate student in my lab, and right now she is a data manager. She is the data manager for the current ENCODE Data Analysis Center, working with me closely on updating the registries and additional annotations. Michael Pacaro was MD PhD student in my lab, and right now he's in his MD phase, very, very close to be done. In a few months, he's going to have both MD and PhD. And he was the person who started the screen project and quite a chief architect behind this tool, even though it seems may not seem very obvious at the beginning, but we have so many elements, so many cell types. It's really a software engineering project, had many challenges. Henry worked closely with Michael, another MD PhD student still in my lab, and he took over Screen after Michael graduated and has added a lot of new features. So he's also a chief architect of the tool. I mentioned the mouse transgenic assays by Lens Lab, and the data I want to mention Diane and Valentina, both of them contributed to generating the data that I showed you today. So this is the cover of the ENCODE paper I already mentioned. So that's all I want to say today. And I thank you for your attention, and I'll be happy to answer any questions.
Thank you, Zhiping. That was great. While people gather their thoughts to ask questions, I just want to make a comment of that I think you really represented well the huge amount of effort that goes into building a consistent and reproducible resource like this. Sometimes people don't really pay attention to it or don't value it enough, but it's really something impressive and of such importance to all of us who want to do reproducible research and be able to compare across cell lines, compare across different organisms. It's a necessary task, and you've taken charge of it and done it in a really incredible way. So I think we're all grateful for that. And now the research can go on and discover novel, interesting things. And Sandro is mentioning he has a question. Go ahead, Sandro. Unmute yourself and that.
Thanks, Elisa. Thanks, Zhiping, for the great talk. I'm curious to know if, for example, have you guys tried, I assume that you have the genome sequence for some of the, at least for some of the samples that you use.
Yes.
So have you tried to associate the lack of a signal to a deletion in that respective genome?
There are some studies by some ENCODE members on that. Yes, yeah, there are a couple papers on that, but for our study, we didn't do it specifically. Yeah, that's a really good point. Sometimes when you don't see a signal, it's because that part of the genome is being deleted. Yeah.
Oh, great. Thank you.
Especially for cell lines, and they can be a bit messed up. And systematic analysis on chromatin interaction by some of the ENCODE groups did find this. Sometimes when you have a matrix, in the matrix, a part is missing, is because that part of the genome is out.
Great, thank you.
Thank you.
Zhiping, I was inspired to analyze data. I wonder if you let people to download, to dump the database, or at least to access by API.
Yeah, that's related to another question by a student as well. If there are any resources on how to download the data from ENCODE and to analyze it, and a tutorial of how to use Screen, if you can point to.
Yes, they are all available. So let me see. Let me drag this over here. So this is the ENCODE portal, and let me close, hide. So this, so I can see. So you can go to, like, you can search Screen directly. And you go to tutorial, and then you can do download. So you can download all of them if you like, or pick the cell type you're interested in, specific ones. And there's an about page that kind of explains it. And it's lots of features. Does that answer your question?
Yes. The access is in screen wenglab.org, right? Is the main
Yes, that's
access.
hosted by my lab. You can get to it from the ENCODE portal.
Easy to go there.
Yeah.
And
There's another question.
oh,
Yeah, you want to finish, Miguel?
no. I'll be back.
Gustavo is asking if ENCODE has anything specific for intron regulators. I don't know if he means splicing regulatory elements or regulatory elements that are contained in introns, which are the majority in humans. But
Yes, there are a set of experiments that the ENCODE has performed on RNA binding proteins. So this is one of the data production. So RNA binding. And many of, just like transcription factors, RNA binding proteins are the regulatory proteins that regulate RNA splicing, stability, localization. One of the ENCODE production centers by Brent Gravely's lab, led by Brent, produces a lot of eClip data and also RNA-seq data after you knock down a particular RNA binding proteins. And those you can get to here. For example, a DNA binding, RNA binding, like here. So eClip data, and those some of the RBPs bind in the intron, some like to bind in the exon, so all kinds. We didn't include them in our registry because our registry is for transcriptional regulation. But if you're interested in those data, you can go and download those.
I'm curious, Zhiping, do you have curated transcription binding sites with experimental data? If I search for transcription binding sites, will I be more closer to Oregano database or maybe to a genomic browser database? How is the quality of the transcription binding site if I'd like to search for one?
Yeah, so our lab actually, Henry, whom I mentioned before, has built this factor book. It's a different resource, but also based on ENCODE data, based on all ENCODE data. And these are the binding regions by transcription factors. So if you want to use that resource, it's all here. And so you can search binding function. So motif enrichment. So these are the ENCODE experiments. And this is the cell line, and that's different cell line. And so this is a motif that's enriched, and you can reverse complement, show consensus sequence, and show different kinds of plots. So for each transcription factor, you get to this page, and all of them are based on, these are all ENCODE data.
And if you are fixed on a gene, if you want to know which enhancer, which promoter is controlling a famous gene,
okay,
is reliable to understand which transcription factors are controlling the expression of that gene.
Yeah, so you can search with a gene. So this is a gene.
Okay. I thought this was the factor that would bind in several places.
So these are the elements. So you can know like the region, which one you want to show, and then you can get these elements, and you can download them.
in GMAP.
Yeah.
But if my question is GAPGH gene for glycolysis, is it quite difficult to build a report for how is its promoter, how is its enhancer, the chromatin domain?
Mm-hmm.
Do you think it's possible to write a report on gene basis, on a regular gene, on
Right.
glycolysis or so?
Yeah, the difficulty is the distal elements. The proximal elements is very easy. And the distal elements, we need to link them to genes. So right now, what we have is whatever data available. So link, let's see, link the genes. So whatever data we have available, but it's not complete. So you know, you can write we can also provide an API to get whatever linked based on existing data, but it's not complete.
Mm-hmm.
That is the difficulty.
Okay. Sure. But I mean, accessing the data, knowing the position of the gene, the genome, I can combine all the data from your tables, right?
Correct. Absolutely. Yes. If you just want to use a distance cutoff, that's easy to do.
Sure. Okay. Let's see if we have more questions from the audience here. Yeah, I'd like actually to work on this proposal with the data to see if we could compare genes, how they are being expressed in different cell lines, and trying to know if there is a special transcription factor in muscle, for instance. I think I can handle the information in order to do that. But that was my question for the database.
Yeah, I should be able to. I mean, the database is targeting people who are interested in individual elements and individual genes, but we have all of the data if you want to do a batch analysis.
Sure, including using artificial intelligence, right? We could try to classify genes that would be potentially more expressed in muscle than in brain, for instance, right?
Exactly. There are some cell types we're particularly interested in. For example, like brain tissues, brain cell types. We're also part of another project called the ENCODE. And that's why we're particularly interested in psychiatric disorders and the brain. And this would be very interesting to compare like brain-specific regulatory elements.
Sure. ENCODE has RNA-seq projects, right?
Yes, ENCODE has a lot of RNA-seq data.
Sure. And,
so
but they are not linked there. Huh? Can we?
Yeah, you can definitely.
Expression data is linked to.
Yes, it's all linked here. So SPI gene expression. This is from ENCODE RNA-seq.
Okay, ENCODE RNA-seq. But you do not do like human proteome atlas that they try to combine expression from GTEC, from Illumina, from other projects.
Not yet.
Not yet.
No, haven't yet.
There's a lot of data on single cell expression. Right?
Correct. Yeah.
This is being very productive today.
Yeah. So that's a really good direction we're going as well. ENCODE is generating some single cell data as well. And as a lab, we have quite a few people working on single cell. That's part of the future.
Sure. You know, in June, we'll be having a talk in this series from the EPI people that do single cells in the atlas.
Yes.
I was amazed that For tissues, they have like ENCODE has some 16, 40 different tissues. For cells, for single cells, they have so many.
Yes.
This area is
That area is
developed a lot. Mhm.
yeah. If you can get the individual cells, the tissue data is just a sum
Sure.
of the individual cells.
Mhm. Some people are concerned about different signatures between different single cells. But as I saw from your data, they are quite conservative, right?
Yeah, it's being very mature now. So a lot of
Mhm.
people, most people generate 10x data, and that's most of the data we see. But there are some kind of combinatoric barcoding, like the Sky approach. They just barcode. It's a bit like the Sprite SIP approach from
Yeah.
the combination of barcodes.
You split and pull multiple times the cells so that by randomness, you will get individual markers for each cell, individual
Exactly.
barcodes after the process.
So single cell is definitely the way to go. Are you working on single cell data, Elisa?
I am. I'm doing a lot of 10x collections for iPSCs,
Yeah.
human iPSCs differentiated in villages. So we grow them all together from different donors
Okay.
so that we can look at EQTLs during differentiation.
Yeah, it's exactly. Wow, it's a very promising area.
So we're trying to link the regulatory elements to each gene based on the perturbations that we see from natural variants.
Yeah, so it's a bit like the CRISPR EQTL, CRISPR QTL.
Yeah, but we don't have to CRISPR because people have SNPs. So
So
there's a group doing the CRISPR too, but we're working with the natural variation.
Exactly. Yeah. So single cell QTL.
Yeah.
Okay, all right.
Right. People are shy or there are no more questions because it was such a great presentation, very instructive. Thank you, Zhiping.
Thank you so much for having me. That was so fun.
It was great. I was quite inspired. I have many ideas. I want to download the database and set up it locally today. Yeah.
Go for it. That'll be great.
I sure will. If I have any problems with the access, I'll write to you, right, to see the. But I see that from your lab, it might be quite easy from that address screen to your lab. I'll try. I'll try from that. That'll be great. So great, Zhiping. You know that we are waiting for using that grant for you to come to Brazil, but I would not recommend it because we have lots of viral variations. The vaccination here is too delayed yet. So because we had plans to bring Zhiping for 15 days to Brazil. It was a program before the pandemic.
Yeah, it's okay. There will be an opportunity.
They are still postponing the grant. They keep postponing. So then maybe someday we'll be able to
yeah, love to visit.
Invite you personally. Yeah.
That's great.
Sure. Okay. So Zhiping, thanks a lot. It was important for us having you for those two seminars to get the year started. It was great that we were already in touch and could organize the list of seminars is now complete with many names. But it was very difficult to get the first ones. So thank you for considering giving us those two talks, and you helped a lot. And
it's my honor.
Yeah, no, it's for us it is. So thanks a lot, Zhiping.
Okay. Take care, everybody.
Thank you.
All right,
Bye.
thank you. So bye everyone.
Ciao,
See you
Miguelito.
around. Ciao, Elisa. Foi doido. Muito bem. Tchau, tchau.
Tchau, pessoal. Até semana que vem.
Vamos terminar a gravação, Thiago? Você já terminou?
Ok, já.